Good morning, dear students. Welcome to your science class. I hope that you all are doing well and reading the chapter as well. Till now, we have done autotrophic nutrition, photosynthesis in plants, and the four components which affect the process of photosynthesis. In this section, we will discuss about the four activities or the four experiments which are given in your book. These experiments will depict the importance of the four components of photosynthesis. So the learning objectives of this section will be the four experiments. The first one is to prove the occurrence of photosynthesis in a green leaf. Second is to prove that carbon dioxide is necessary for photosynthesis. Third, to prove that chlorophyll is necessary for photosynthesis. And the last one, to prove that sunlight is necessary for photosynthesis. So here is our first experiment, which is to prove the occurrence of photosynthesis in a green leaf. One thing I would like to tell you before starting the experiment, that in all the four experiments, we will be testing the presence of starch. Now what is starch? Starch is a complex form of glucose. As we all know, we have seen earlier also that during the process of photosynthesis, glucose is synthesized, and this glucose is used up by the plant for its life activities, and the rest of it gets stored in different parts of the plant in the form of starch. And to test the presence of starch, a special chemical is used, which is iodine solution. And the characteristic of this chemical is that whenever it is added to starch, it turns it into blue-black color. So in this first experiment, what we have to do is, first we have to take a potted plant and then it is to be kept under sun for a few hours. Why it is kept under sun for a few hours? So that it can perform photosynthesis, it can synthesize glucose, and starch will be present in the body parts of the plant, especially in the leaves. Then after this, a green leaf from this plant is taken and then it is put into a test tube, which is there in this diagram, and in this test tube alcohol is filled. Why alcohol? Because alcohol takes out the chlorophyll content from the leaf. Now this test tube in which alcohol is there and the green leaf is there, it is then kept in a beaker which is half filled with water, and this whole setup is put on a burner. Means we have to boil this. Now mark it, actually we have to boil the alcohol with the leaf, but we can't boil alcohol directly on flame. Why? Because it is inflammable. So we have to prepare a water bath for it so that it will be safe to boil it. Now after boiling, we have to take out the leaf from the test tube using a forceps. Never use your hand. And then keep it on a dish. Then put a few drops of iodine solution on the leaf. Before putting iodine solution, wash the leaf thoroughly with water and then put it on a dish and then put a few drops of iodine solution. What will you observe? As I have told you earlier that iodine solution turns starch into blue-black color. So you will see the leaf turning blue-black in color. So it proves what? It proves that when iodine is added to starch, its color changes to blue-black. Thus change in color of the leaf to blue-black on adding iodine solution indicates or proves the presence of starch in it. And how the starch can come into that leaf? It is because of photosynthesis. I hope the first experiment is clear to you all. Now next experiment is to prove that carbon dioxide is necessary for photosynthesis. So it means that we have to prove its presence is important for the process of photosynthesis to occur. So to prove its importance, we have to make carbon dioxide absent for this process. Now how can we do this? For this we have to take the help of a special chemical, a magical chemical which is called potassium hydroxide. Why potassium hydroxide? Because it has the ability to absorb the atmospheric carbon dioxide. It means wherever we'll put potassium hydroxide, it will absorb all the carbon dioxide present in that particular area. So let's start the experiment. What we have to do is to take a green potted plant. Then we have to take a petri dish. A petri dish is a small glass dish. In this dish, we'll put some potassium hydroxide and place it near the potted plant. Now we have to cover this whole setup with a glass bell jar. Now bell jar is, as the name indicates, is bell-shaped glass jar. Then we have to put this whole thing under sunlight so that it will have all the factors for photosynthesis. That is, the plant is having chlorophyll, the plant is having sunlight, the plant is having water. But what is absent? Carbon dioxide. So we have to keep this whole setup for a few hours in sunlight. Leave it there for a few hours, and then after few hours take out the bell jar and take one leaf from that plant. In this picture I have drawn the leaves yellow in color, which is showing that photosynthesis has not taken place. Leaves have not prepared their food, and that's why they are turning pale yellow. Now take out one leaf from this plant and test it for starch. You know how to test it for starch, as we have done in the first experiment. Put the leaf in a test tube filled with alcohol, and then put this test tube in a beaker half filled with water and keep it on a burner. Boil it, then after some time take out the leaf with the help of forceps, clean it with water, and then put it on a dish. Then add a few drops of iodine solution. You know that iodine solution turns starch into blue-black color. So what should be the result in this case? Yes, after putting iodine solution, there will be no change in color. Why? Because there was no carbon dioxide in the bell jar, so the plant was unable to synthesize glucose and hence no starch. So by this experiment, we have proved that carbon dioxide is necessary for photosynthesis. So to conclude, we can say that the leaf from the plant in the bell jar with potassium hydroxide will not have any starch, and this shows that carbon dioxide is needed for photosynthesis to take place. Our next experiment is to prove that chlorophyll is necessary for photosynthesis. Now for this experiment, we have to take a different plant which will have variegated leaf. Now what is variegated leaf? Variegated leaf means a leaf which is having scattered form of chlorophyll. In this leaf you can see in the middle the chlorophyll part is there, and at the outer side it is yellow in color. So such type of leaves are called variegated leaves, and the example for them are coleus and croton. These are very common ornamental plants and you can easily find them. Now what is to be done? The same thing, we have to take one leaf from such plant, put it in alcohol and then keep the test tube in a beaker filled with water, then boil it for 10 minutes and then take out the leaf with the help of forceps, clean it with water and place it on a dish. Then we have to put a few drops of iodine solution to the leaf, and then what should be the result? In this leaf, the parts which are having chlorophyll, means those parts which were green in color, they will turn blue-black, while the other parts which were yellow in color, where the chlorophyll was not present, there will be no change in color. Okay, can you see from the picture? In this picture, the middle part has changed into blue-black color. Why? Because the middle part of this leaf was having green color. It means this part was having chlorophyll. So this part has changed into blue-black color and rest of it is same. So hence it is proved that chlorophyll is necessary for the process of photosynthesis. So to conclude, we can say that the green parts of the leaf, they have synthesized starch due to the presence of chlorophyll, and thus they turned blue-black when iodine solution was added to it. Hence, it proves that chlorophyll is necessary for photosynthesis. Our last experiment is to prove that sunlight is necessary for photosynthesis. Now to do this experiment, what we have to do is first we have to keep a potted plant for 2 days in dark. Why we are keeping this plant for 2 days in dark? So that whatever starch or glucose is present in its body, it will be utilized within 2 days when it is not performing photosynthesis. Then after 2 days, we have to take out the plant and take one leaf, not to take out from the plant, but it is still there with the plant. One leaf and its middle part, cover it with black chart paper so that it does not get any sunlight. And then keep this plant in sunlight for a day. Then again do the same starch test what we have done in the previous 3 experiments. Take out that leaf. After few hours, take out that leaf where you have put the black chart paper in the middle. Then take out the chart paper and do the starch test, whatever you have done previously. And then in the last step, put the leaf after washing with water on a dish and put a few drops of iodine solution to it. What will happen? You will notice that the part of the leaf that was deprived of sunlight, where you have covered with black chart paper, where sunlight was not there, that part, by adding iodine solution, turns into yellow-brown color, while the other part, which was green, it turned into blue-black color. Why? Because that black part was not having sunlight and therefore it was unable to synthesize glucose, while the other part was free, it was exposed to sunlight and it was performing photosynthesis and therefore synthesizing glucose and starch. Thus, the blue-black color of the exposed part of the leaf is due to the presence of starch in it. And the absence of starch in the middle part or the covered part results in yellow-brown color. So to conclude, we can say that the absence of starch shows that photosynthesis did not take place in the part of the leaf that was deprived of sunlight. And this proves that sunlight is necessary for photosynthesis. So these are all 4 experiments, and I hope that you have enjoyed learning these 4 experiments. Please go through them. These are there in your book, and I will give you the written notes of these experiments which you have to copy in your notebook with the diagram. Thank you.